Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Alex Nicolaitis, the webcast coordinator for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the Q&A window on your screen. After the presentation, we'll begin the question and answer portion, and I will ask our speaker your questions. Your questions in the Q&A window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Corinne Branciaroli, Group Leader, Viral Cell Line Development from Oxgene, a WuXi Advanced Therapies company.
Hi, everyone, and thanks to BioProcess International for the introduction and for hosting our talk today. My name is Corinne Branciaroli, and I've been working at Oxgene for more than 5 years. Today, I will introduce you to the development work that Oxgene has been carrying out to establish a technology of packaging and produce stable cell lines for lentiviral vector manufacture. The 2 items on the agenda are introduction to our company, Oxgene and WuXi Advanced Therapies, and then we will move on to LentiVEX approach and explore transition from transient to stable cell lines for lentiviral manufacture. Oxgene was founded in 2011, and we became part of WuXi Advanced Therapies and WuXi Aptech family in 2021. Together, WuXi Advanced Therapies and Oxgene offer a seamless end-to-end solution to support and accelerate the development and manufacture of cell and gene therapies. We aim to establish long-term partnerships with our customers, and we can support them all the way from discovery research to eventual commercialization of the final product. In particular, Oxgene specializes in the discovery science and preclinical research, and WuXi Advanced Therapies can take the customers through the latest stage of development and manufacturing steps for clinical trials, testing, but also regulatory approvals and commercialization. So, just to give you a bit more context, the work subject of this webinar has been developed thanks to the cell line engineering capability at Oxgene. By researching a new technology, we move on to development and establishment of new platforms. Our products can then be onboarded into manufacturing processes for the desired viral vector. But you might ask, why cell line engineering? I believe that we are all familiar with the challenges that are presented by the lentiviral vector manufacture, such as scalability, cost, and variability. So, by using a cell line engineering approach, Oxgene aims to provide a solution that can reduce costs and variability while increasing or maintaining good yields in upstream process. So if we look at the CLD campaign on workflow displayed on your screen, we started by looking at the transgenes that we wanted to insert into a host cell line. Then we stably integrated these transgenes and selected the stable pools, which stably retained this functionality. We then moved on to single cell sorting to identify clonal cell lines, which were harvested and expanded and submitted for several rounds of screening so that we could identify the top performer showing the desired characteristics for cell line banking and to support the lentiviral vector manufacture. So before embarking onto the development of such cell lines, let's take a look at what we knew and what we didn't know in year 2017. At that time, we certainly knew that the industry standard for lentiviral manufacture was the 4 plasmid transient transfection, which requires 4 different plasmids, which carry the packaging elements, envelope, in our case VSVG, Gagpol and Rev, as well as the lentiviral transfer plasmid, into a HEC2 and 3 cell line. This process reported good yields, but also high costs and variability. So, if we were to move toward stable cell lines and integrate the packaging elements, VSVG and Gagpol and Rev, into a packaging cell line, or even take a step further and also integrate the lentiviral transfer plasmid to make a producer cell line, what would this mean? Certainly, we would cut down in costs of the plasmid manufacture because the packaging cell line requires only a single plasmid transfection plus induction, while the producer cell lines require no plasmid transfection at all, only induction. Our technology allows to resolve the cost challenge, but what about variability and yields? Well, these were indeed the questions that Oxygen wanted to answer, and our work in the past 5 years is what allowed us to provide answers to these questions and is the subject of this webinar today. So I will proceed to show you how we addressed those questions. This is a brief timeline to help you put things in context, and I will now explain each step of the process in more details. How did we start? We started with plasmid engineering, of course. The LentiVEX plasmid kit is a third-generation self-inactivating packaging lentiviral vector system. Our molecular biologists and virology experts used codon optimization and promoter screening approach, aiming to maximize the yields, and also reduced homology to maximize safety, transiently yes, but also with the idea of a stable integration in mind. We also adopted a new HEC2 and 3 suspension cell line, and when our plasmid set was tested against commercially available lentiviral vector kits, we observed good yields and good physical to infectious titer ratio. So the packaging elements, the plasmid sequences overall, and the cell line seem to be a good starting material for the stable integration. But here's the question: can it work? The first step was to integrate 2 packaging elements, envelope VSVG and Gagpol, under the control of a tetracycline regulated promoter into HEC2 and 3 suspension cell line. This was a preliminary stage of clonal cell line development, and we refer to it as pre-packaging cell line that you can see displayed on the left side of your screen. We then carried out a second integration containing Rev and lentiviral transfer plasmid to make a producer cell line, which is on the right side of your screen. We used GFP, green fluorescent protein, as gene of interest. We went through a CLD campaign and identified the top performing clonal cell lines. As you can see from the graph on the left, the 4 producer clonal cell lines were showing titers of approximately 2 to 3 E6 TU per ml. So the producer proof of concept confirmed that the approach was feasible. The stable cell lines were able to retain functionality of integrated transgenes and produce lentiviral vectors with reproducible titers among the two runs. And yet, as you can see on the screen, on the graph on the left, the 4' plasmid transfection protocol had been optimized by that point, so now we were getting higher titers with the 4' plasmid transgene transfection. Therefore, the gap in productivity between the producers and the industry standard was still high. And here the next question comes: how can we improve it? Well, the question was partially answered by the next stage of work we were carrying out in parallel to the producers. The stable integration of REV, third packaging element, into pre-packaging cell line to generate a packaging clonal cell line. After another round of CLD campaign, the top two clonal packaging cell lines, in light blue in the graph on the left, were compared with the pre-packaging cell line in gray, and we observed higher yields with the new products, in particular with product LB001. Our idea at the time was to propose this packaging cell line as lentiviral vector manufacture platform of choice, using a simple lentiviral genome transfer plasmid to be transfected transiently. This will certainly give us reduced costs, 1 plasmid instead of 4, and also flexibility to use with this platform with several transfer plasmids carrying different gene of interest. But although within the same experiment we observed reduced variability compared to the 4 plasmid transfection, by monitoring the platform for 1 year, we discovered that the platform still presented variability of lentiviral vector titer at similar level to transient transfection. And among all the factors, such as operators, lab facility, cryovials, and cryobanks transfection protocols, we identified a strong correlation between transfection efficiency of the lentiviral transfer plasmid to lentiviral vector titer productivity. What were our findings so far? On the producers, we knew that the proof of concept worked, and starting from the same pre-packaging cell line, integration of the REV and lentiviral transfer plasmid resulted in titers approximately in the range of E6 TU per ml. On the other side, the packaging cell line proof of concept also worked and showed a higher productivity compared to the pre-packaging and as well to the producers. So, what was the limiting factor for the producers? We carried out an experiment to transiently add excess of various packaging elements and lentiviral transfer plasmids, and we found that this latter one had the highest impact on titer. Here's the next round of questions to address. Question 1, how can we increase the integrated copy of lentiviral genome carrying the gene of interest? And question 2, can the packaging cell line be a better substrate than the pre-packaging cell line for producers? The next stage of development, we used the packaging cell line as starting cell line platform and a transposon-based system to aid multiple copies integration of lentiviral transfer plasmid into our version 1 producer cell lines. This approach showed significant improvement in titers, up to 20-fold higher than the first proof of concept of our producer cell line generation. And after another round of CLD campaign, these producer clonal cell lines showed titers as high as 1 to 2 E8 TU per ml. The graph on the right shows three different productions carried out between passage 10 and passage 20, and the four different bars for each production are three clonal cell lines and the pool they were derived for. We understand that all this work was carried out using GFP as gene of interest, and this is a rather small transgene, easy to express for the cells and also well tolerated. Hence, currently, we are trialing the approach with therapeutic gene of interest, such as the not limited to Cas19. As anticipated before, Oxgene and Wuxi advanced therapies are all about continuous development. And thanks to the acquisition and expanded cell line development capabilities at Wuxi Advanced Therapies, we identified a new cell line platform able to double titers of lentiviral vectors when compared to our original HEC2 platform version 1. This new platform version 2 will be the perfect substrate for further engineering, and LentiVEX packaging and producer cell lines version 2 are currently under investigation. To conclude, the 4-plasmid transfection remains our industry standard. The team at Oxgene worked hard to answer these questions about variability, yields, and cost of goods from the different platforms, and we demonstrated how packaging and producer cell lines can help to overcome upstream process manufacturing challenges, reducing the costs and variability, but also maintaining high yields. Overall, the LentiVEX technology allows flexibility and easy transition from transient to stable cell lines platforms. Thank you very much for your attention today, and I'm now happy to take any questions.
Okay, great. Thank you. As a reminder to our audience, you can go ahead and type in your questions for our speaker now. So our first question is, what stage of development are you at currently?
Oh, thank you for your question. And what I can say is that we are toward the end of development, meaning that we are ready to work with our customers to identify the best platform to suit their needs. In terms of transient platform, between Oxgene and Wuxi Advanced Therapies, we have an established process for both research and clinical manufacture. In terms of packaging cell lines, we are working toward GMP banking and process development of the packaging platform version 2 that we want to use as a packaging platform on its own. And regarding the producer cell lines, the development service of making a custom producer cell line can be offered with our version 1 platform because it demonstrated it can achieve very high titers comparable to the industry standard of the 4-plasmid transfection. These products are also available for customers to receive our research-grade products. So, for example, the producer cell line containing eGFP as GOI, and they can test them in their facilities.
Great. Our next question is, your stable cell lines adopt a tetracycline regulated system. Did you check for basal expression? If yes, what's the range?
Oh, this is a good question. Yes, correct. The packaging transgenes are regulated by a tetracycline regulated system. And yes, we carry out a non-induction control which shows normally undetectable values, or sometimes the highest that we've observed is in the range of E4, E5 TU per ml. To this, I also want to add that during our cell line development campaign, we monitor the regulation of the system. So we test the cell lines in presence and absence of inducible agent, and the titer regulation is one of the selection criteria. So we would choose a cell line that presents the lowest, the lowest in expression, or like no expression in absence of induction agent, and the highest expression in presence of induction agent. I hope this is clear.
So it looks like we're running short on time. If we don't get to your question, we'll be passing them along to Corinne, and she can follow up with you directly. So please go ahead and continue to type in any questions you have, and we'll get those passed along. The last question for the webcast is, in this presentation, you express titer in TU/mL. How is this determined?
Yes, so that is the infectious titer transducing units per ml that we carry out by transduction of hectin and 3T adherent cell line, and the cells after transduction are incubated for 3 days, 72 hours post transduction, the cells are harvested. And in this specific case, as we are using GFP as Lentivirus, we analyze the transduced cells by flow cytometry for detection of GFP signal.
Okay, well, thank you, Corinne, and thank you to our audience for joining us. The recorded version of this webcast will be made available for on-demand viewing on our website, and as a registered attendee, you will receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.